Introduction to Bioinformatics. Now, these interactive sessions are optional, and we really encourage you to go through the synchronous materials, but we'll also record these sessions so that everyone will have access to the recordings after the session is over and can follow up on the different assignments that we do together, gaining hands-on experience. Sorry. So remember that in total, we will have 6 interactive sessions where we will discuss different aspects of analysis of DNA, RNA, and other types of next generation sequencing data. And we'll discuss a few different hands-on projects that you can work through to really get an introduction to bioinformatics, including some aspects of working with data using some big data tools and working with sequences and different process data sets in R and Python. Now, the coding portion here is again optional. The introduction to bioinformatics is primarily going to be for those of you that come from a biology background and are interested to understand how you can actually apply the different methods on different types of data. So last time we talked about how this program will include different types of asynchronous resources, including the processing server, where you can run big data analysis, and we'll do some demos today to make sure that we understand how genomic data is analyzed. And also we have a number of introductory courses that are available on the educational portal. To understand how everything is kind of connected together, you will find a syllabus on the organizational page. So let's take a look at the organization page just to review what is posted there in terms of courses and assignments and the syllabus. So this is the link. Just put this link in the chat. Okay, so this is the organization page. Thank you, Vipsa. On the organization page, you will find today's session, which is Introduction to Genomics, DNA Variants and Mutations. So we'll talk about this today. You'll also see right here different posts from activity, different participants that have been completing coding assignments. Irmak has posted a question on the forum that we will address today. And we will find here courses. So on the forum, you will also find the recordings of previous sessions and different types of activity. If you have a question, that's the place to put your questions. If you have a, if we have an announcement or we have any additional resources for you to go through in the course of the program, we will post that here as well. Here on the Courses tab, you will find some of the coursework that is included, and this is going to be according to the different topics that we cover in our interactive sessions. So for example, last time we asked you to start working on the Introduction to Bioinformatics course, and today we'll go through Introduction to Genomics and Genomics 1. You can also see the full syllabus of this program right here. If you go to the menu and go to the syllabus tab. So the syllabus tab explains how the interactive sessions and the topics that we cover there are connected to the online courses. So for example, here you can see Bioinformatics and Big Data Concepts and Applications. What's the course associated? Introduction to Bioinformatics. Today we're talking about genomics. What is the associated course? Introduction to Genomics and Genomics 1, and so forth. So really, this is your central place to understand where we are in the program, where we are going next, and how you can best leverage the other additional resources that we have available on the educational portal and the platform to benefit the most from this course. So let's do a little review of what we covered last time. Last time we talked about the need to process and analyze high throughput data sets and talked a little bit about the logic of building bioinformatics pipelines. We also talked about this project-based learning approach for bioinformatics, where you can direct your activities into the topics that are most interesting to you. And so that is something that we will try to address today as well. We'll give a few different examples, but it's up to you to be able to understand where to find the different resources that can guide you in that direction. Now, if you have any questions about this, it would be a great time to ask about this the different types of data sets and projects that are available at the end of this session. We also talked a little bit about how from getting to understand the logic and being able to handle big data enables you to focus on one of the more interesting aspects of bioinformatics, which is data science and biological interpretation. It's not a must to know both, but we've talked about how biology is acquiring the characteristics of data science because there's so much data being generated in life sciences. And contrary to what it might seem like when we see all of the amazing articles that publish cutting edge research and all of the findings, we are still very far away from fully understanding how cells function and how the world is also functioning at the molecular levels. So this high throughput data contains a lot of novel information and novel insights that are yet to be discovered. And so we're hoping that you're also going to become one of the people that discovers some of these important aspects of molecular biology by leveraging data. One of the places that you can start with by going through this introduction to bioinformatics course is really understanding how bioinformatics fits in to various disciplines and domains. For example, in the introduction to bioinformatics course, we discuss how bioinformatics is used in biomedical research, biotechnology companies, pharmaceutical companies, clinical research organizations, and we also talk about other applications in agriculture, even defense and security, and other types of domains. So it's really a starting point for you to get to know bioinformatics. It's also a starting point for us to get to know you. And so that's where we hope to understand your level of education, maybe what is your current institution that you belong to, and any other type of information that you would like to share. Ultimately, what we are going to do with this information is we are trying to build a community that can work together on different types of projects leveraging bioinformatics. And so it's important for us to understand the level of different participants of different programs and try to create the best and most effective path for those of you that are learning bioinformatics to become an advanced user or even a developer of different novel bioinformatics methods. Now, previously, we still ask today for those of you that complete the courses to give us feedback, and it's important for us to understand that the materials that we put out and we constantly update are also useful not only for students but also for faculty. And what we've seen is that there is a surprising amount of depth and a good starting point for anyone that's interested in bioinformatics. What this course talks about is the increasing availability and even an exponential growth of data in biology and life sciences in general. And as a result, the computational advances from the hardware standpoint as well as the software standpoint, are really driving biological research to allow us to study biology at the level of subcellular regulation, as well as the variety or heterogeneity of different biological phenomena as we're able to grasp and explore insights from this data. So it is a lot of data to manage, and there are many different types of data to integrate. And I think that's one of the challenges as well as the opportunities of bioinformatics. Now, last time we also talked about how in the context of biomedical bioinformatics, or bioinformatics applied to biomedical research, there is an exciting application that includes the integration of multiple types of omics data. And that also brings us to the point of integration of multiple types of informatics, bioinformatics, chemoinformatics, clinical informatics. So really, it's an opportunity to look at it from a holistic perspective, understanding some of the applications in precision medicine and biomedical research. And so as we go through the program, we'll continue to include a number of different examples that illustrate these types of aspects of biomedical informatics or bioinformatics in biomedical research. If you already started or completed the Introduction to Bioinformatics course, you will see that in this course, there are three major segments: Introduction to Big Data Bioinformatics, which really is a new thing in some sense, new in the sense of the last decade. Bioinformatics has been around for a long time, and there has been a tremendous kind of a dynamic to this field. So there's a lot of new ideas and new approaches that are being developed as we speak, and therefore the term itself, bioinformatics, is changing as well. So as we go through this course, we'll try to address some of the traditional aspects of bioinformatics as well as some of the newer aspects of bioinformatics that are important to know for those of you that are going to continue to apply bioinformatics in different domains and will try to master bioinformatics in your research. The other course that we recommend to start going through is the Introduction to Genomics, even though we will cover some aspects of that course in today's session. Okay, so last time we also had an assignment, and so these are the several things that we asked you to do. Complete your profile on edu.tbioinfo, which means to add your full name, to explore the different resources like this page that I mentioned for the organization, and hopefully make your profile discoverable. So to make your profile discoverable, you have to complete it with your full name, add an avatar, maybe post some social links that people can find you on LinkedIn or ORCID or some other social network. We also asked you to explore the server.tbioinfo, and if you're interested in coding, code.omicslogic.com, and I'm excited to see that many of you have started that process as well. And also to complete the Introduction to Bioinformatics course that includes completing that survey at the beginning of that course. So it is great to see that some of you have taken the opportunity to do so. J.Deb Powell, who's been very active, already completed a couple of courses in his profile. William Guesdan also probably has participated in other programs before, but has been making some progress on his coursework. And Prashant Misra, who also just joined and has completed Introduction to Bioinformatics. So for all of you that have yet to complete your profile, we have been commenting on your activity, as well as BIPSA will be reaching out to you directly to try and help you complete your profile and make sure that you understand where you can do that and how it's useful for your experience here. Now, I also wanted to ask that all of you who are interested to become a part of this bioinformatics community also join our social media group. So we have a group on LinkedIn and a group on Facebook that you can join. And those groups really help us share upcoming information about upcoming programs, the different resources, job opportunities, and exciting research that is relevant to the different programs that we run. And we run programs on genomics, transcriptomics, metagenomics, epigenomics, machine learning for biomedical data, as well as specialization tracks like bioinformatics for infectious diseases, bioinformatics for oncology, and precision medicine. Okay, thank you, BIPSA. BIPSA posted the links there. So if you're a part of any of our programs, we welcome you to join those and interact with other participants that we have from previous programs, from industry, and from academia. So today's session will be focused on the introduction to genomics. How do we understand DNA variation or DNA variants and mutations? And we'll talk about it in the context of next generation sequencing. So we'll talk a little bit about what we can do with next generation sequencing data, and what are some of the different methods of analysis that you can leverage to study this data and understand important variation, so variation that is significant for some kind of phenotype. We'll try to go over the shotgun sequencing challenges with mapping of short reads, the need for structuring data so that it is useful for downstream analysis, different types of variation, and different types of locations for those variants that are important for interpretation. So what we'll do today is we'll cover one of the sections on the T-Bioinfo platform that's called mutation variant, and we'll see a few examples there. And we will also use some additional software like JBrowse, and I'll introduce you also to how to install and configure the integrative genome viewer that is going to be useful for those of you that are planning to complete Genomics 1. So if you need some of these links, I'm just going to paste that in chat as well. So obviously server.t-bio server-t-bio.info is one of the places that we will use, and another one is going to be this integrative genome viewer. And for those of you that are going to go through the Genomics 1 course, we ask that you also install, and today we'll hopefully configure it so that you can use it later. Okay, so what is covered in this Genomics 1 course? So this course talks about some of the basics about DNA, point mutations, and other types of mutations like multinucleotide variation, including rearrangement, insertions, and deletions, or DNA aberrations. We talk about different types of genomic information based on sequencing protocols, and some of the challenges with interpretation and analysis in application to different examples that we'll talk a little bit about today. Okay, so let's talk about DNA, or the code of life. First of all, let me ask you a question. How many chromosomes do we all have? Okay, excellent. So 23 pairs or 46 chromosomes, as we can see right here. And so what we know about DNA is that DNA molecules are tightly packed around proteins called histones to make structure called chromosomes. So chromosomes are really DNA wrapped around histones. Humans have 23 pairs of chromosomes in every cell, so that is 46 chromosomes in total. The sex chromosomes determine gender, so XY is male and XX is female, and other chromosomes are called autosomes. Who knows what is the largest chromosome? Which chromosome is the biggest one, the longest one? So a hint, you can look on the screen and guess from there, if you don't know. All right, thank you, Prashant. Yes, it is indeed chromosome 1. So how many genes does chromosome 1 contain? Yep, exactly. Irmak, based on the figure, first chromosome, that's true. Okay, so the first chromosome contains about 8,000 genes. The smallest chromosome, chromosome 21, contains about 300 genes. And in fact, there's a kind of a funny story there. Chromosome 22 should actually be called the smallest, but the scientists made a mistake when they first numbered them. So the number of chromosomes actually correlates with their size. Okay, so DNA itself, genes are made of a chemical called DNA, which is short for deoxyribonucleic acid. And the DNA molecule is a double helix, of course. So these are two long, thin strands twisted around each other like a spiral staircase. The sides are sugar and phosphate molecules. So how many nucleotides are there in the human genome? Excellent. Yeah, so Irmak spilled the beans. There's about 3 billion base pairs. So there's about 3.2 actually base pairs. And a lot of that information is actually not protein coding genes. So let's talk about some of these numbers. So in humans, genes vary in size from a few hundred DNA bases to more than 2 million bases. And as we know, the annotation of this information of the different genes was started by the Human Genome Project. So when scientists started the Human Genome Project, they were expecting to annotate about 100,000 genes. How many genes do we know of today? Who knows? How many genes are there in the human genome? Okay, any other guesses? How many genes are there in the human genome? Okay, so Prashant is saying 30,000, Irmak is saying about 20,000. Okay, Aruna is saying 30,000. Okay, so the primary goal of the Human Genome Project, which lasted between the 1990 until 2003, so it's 13 years, was to determine both the DNA sequence and the location of the estimated 100,000 human genes. So scientists at the time, if you think about it, 1990, that's just 30 years ago. So at the time, scientists believed that once we have the full sequence of the DNA, we would fairly quickly be able to determine where all the genes were. But as we know today, it is actually not clear how many genes we have. So today there are several competing human gene databases with many thousands of differences among them. And although the number of protein coding genes has gradually converged at about 22,000, maybe to 25,000 genes, the number of other gene types has exploded. So here you can see some of the information that is contained in the human DNA. You can see that about half of the DNA are what's called defective transposons and fragments, which is called the DNA dark matter, or junk DNA as it originally was defined. Now, also surprisingly, the humans are not the most, do not have the most genes. For example, some of the plants like grapes have over 30,000 genes. And you can also compare that to simple organisms like E. coli that have over 4,000 genes. So even though the difference in size and complexity might be dramatic, the number of genes is actually comparable, right? So it's only about 5-6 times less than the humans. Now, the DNA itself is comprised of these molecule chains that are working together, and they form what's called bases, base pairs, and nucleotides. So when we say 3 billion or 3.2 billion base pairs, what do we mean? So in between the outside strands of the DNA are pairs of chemicals called nitrogenous bases, or bases for short. And there are 4 types of bases: adenine, A, thymine, T, guanine, G, and cytosine C. These bases are linked in very specific ways. A always pairs with T, and C always pairs with G. The genes on the DNA are encoding for RNA. And so we know that the central dogma of molecular biology is that DNA is transcribed to mRNA and mRNA is translated into protein. Again, this process is only relevant for these 20 to 25,000 genes, not for all of the different elements that are in the DNA. But one of these important processes is that process of transcription and translation, which happens in trinucleotides. So trinucleotides are called codons, and they change in one of those nucleotides within the codon is actually what we call a variant, a DNA variant. So what types of DNA variation is there? One big type is called point mutations. So these point mutations mean that there's one single nucleotide that has changed, but there are different implications for the different types of changes. For example, there could be actually 5 types of changes for each one of the nucleotides. Why 5? So the first one is for each one of our nucleotides, we can have 3 different substitutions, right? So for example, if we have a total of 4 different nucleotide options, and it has to be one that has changed, right? We're left with 3 alternatives. The other 2 alternatives are insertion and deletion. And so out of all of these 3, which one do you think has the most impact on the protein that is encoded within that gene? Okay, so both Prashant and Susanna have answered insertion and deletion, or deletion. So let's take a look. If we have a substitution, oftentimes we are just talking about one single codon changed, so that will be one amino acid that has changed. And in fact, insertions and deletions can lead to a change in subsequent sequence of the whole amino acid. And so one of the differences is that we oftentimes have a variety of levels on how much impact that it has. Because if you think about a protein, protein translation can stop because of what's called a stop codon. So one of these nucleotide changing the sequence can lead to a stop codon. And it can also change the whole subsequent sequence, making the protein shorter, or even changing it completely, so introducing different types of changes. Now, as we think about this, we also have to think about how to classify these different mutations. So the way that they have been classified, these single point mutations, are what's called missense mutations, nonsense mutations, and frameshift mutations. So a missense mutation is a change of a single DNA base that results in a change in the amino acid sequence. So sometimes one single amino acid change can greatly alter the protein's function. For example, we'll talk about cystic fibrosis. That is that kind of a mutation. A nonsense mutation is a change of a single DNA base that creates a stop codon, which terminates translation. So as a result, you have a shortened protein that may not function or may have an abnormal function. And finally, we have this frameshift mutation that results from the addition or removal of DNA bases that shifts the DNA sequence and the corresponding amino acid sequence. And the result is a protein whose sequence, structure, and function are very different from those in the original proteins. Now, to study this kind of variation, we need to understand exactly what is in the DNA. And one of the challenges that relates to bioinformatics is the analysis of sequencing data that allows us to piece a huge puzzle of information together and identify some of these changes at the level of DNA. So we think that that will eventually change the way that the proteins are produced, and that's why it's important to understand exactly what that change is. So what are some of the methods of sequencing that we have to be able to study this kind of variation? If we just take a look at Illumina sequencing, or Illumina actually adopted just one of the sequencing technologies. Originally, there was Sanger sequencing, and Sanger sequencing remains a gold standard for accurate identification of DNA variation. However, Illumina has proven that the shotgun sequencing approach is both accurate enough and computationally easy to analyze, easy enough to analyze for it to become the de facto standard in sequencing. So we can think of 3 different options that this kind of sequencing produces: whole exome sequencing, whole genome sequencing, and what's called targeted sequencing. So when we talk about whole exome sequencing, we're talking about sequencing just those 20 to 25,000 genes. And when we talk about whole genome sequencing, that means that we are sequencing everything that is in the genome. So as you can see, there is a huge variation in just the amount of data that those two approaches produce. And that's why sometimes people have to sacrifice coverage, and we'll talk about what coverage is in just a second. They have to sacrifice coverage of each particular position on the DNA to gain a perspective on the whole genome. With whole exome sequencing, there's still some limitations as to how much it costs and how much data it produces to sequence all of the 20 to 25,000 genes, because even though that's a much smaller number, that is still a pretty big number of nucleotides that have to be covered. And the final approach, an approach that is most widely accepted for clinical applications, for example, where identification of a mutation has serious clinical implications, is what's called targeted sequencing. And targeted sequencing means that you can have high coverage of that particular short sequence, maybe one or a panel of genes. And as a result, you have a very clear idea of what types of mutations are present and in which proportion. And we'll talk about that in just a second. So as a result, we have targeted panel sequencing. For example, 40 to 400 genes is a typical approach. It is high coverage, it is manageable cost, and typically used in clinical or very specific clear situations. Whole exome sequencing is approximately 20,000 genes, 20 to 25,000 genes, intermediate coverage. It allows us to study variation in many genes at the same time with lower coverage. So a lot of this has an ability to show us important information about some more ambiguous situations in biomedical research. And then whole genome sequencing typically is in the domain of exploratory analysis, where we want to understand what type of whole genome interaction or variation is associated with maybe a phenotype, or we are trying to understand some basic biology behind what is in the DNA. Now, obviously, here we're talking about human genomes, but sequencing is also applied to other types of organisms. So, for example, we can talk about metagenomic and viral sequencing. So, also applicable to find variation, but very different type of variation. For example, when we talk about targeted metagenomic sequencing, we're actually looking for taxonomic variation. We're not talking about how is a genome of a particular, for example, bacteria different from the other similar types of bacteria. We're a lot of times looking at, for example, metagenomic sequencing of a microbiome. And that allows us to understand differences in composition and characterize that composition by taxonomic organization. We can also sequence total DNA or RNA and amplify for a particular type of an organism, for example, one type of a taxonomy or one type of a virus. And as a result, we can study how these viruses are different. And so there are these different types of approaches that allow us to look for emerging mutations, for example, mutations that are responsible for antibiotic resistance. It can help us understand emergence of novel pathogens or help us study the microbiome to understand its composition. Okay, so let's talk a little bit about data analysis. So first of all, many different types of sequencers exist, and they basically divide up into two categories: the short read sequencers that we talked about today, and long read sequencers like PacBio and Nanopore, Oxford Nanopore. So long read sequencers are making amazing advances in the length of a read. For example, PacBio recently has developed hundreds of thousands, 100 to 150,000 long read sequencing, which allows us to study in an unprecedented precision those areas that have a lot of repeats, for example. So as we saw in the chromosomes, there are huge areas of repeats that are impossible to map with shortcut sequencing. So now as we reanalyze the human genome, these long read sequencers are playing a major role in that. On the other hand, Oxford Nanopore has much lower precision per nucleotide, but allows us to study whole genomes of individual bacteria and even human samples, but at a much lower cost. So whatever way we go, with whatever type of a sequencer, we're going to be generating a lot of data. So let's think about what this data contains. First of all, there are multiple formats that sequencing information is stored in. For example, the FASTQ files provide us with reads from the sequencer. It is a text-based file format that stores nucleotide sequences of reads and quality scores for each letter. FASTA is a text-based format for representing DNA and protein sequences, even though some machines produce a FASTA for reads. As you can see right here, these are individual reads in a FASTA format. And then finally, a typically leveraged type of data is stored in GTF format, which is called genome transfer format, and that includes the beginning and the end of each genomic element. So if we have exons or introns or full genes and transcripts, those will be included. So what is the difference between GTF and GFF files? Actually, there's no difference, they're exactly the same. So GFF is the more advanced, it includes a little bit more information in it, and GTF is the standard. So a lot of GFFs could easily be converted to GTFs. So here we see FASTA files. For example, in FASTA files, you can store full genomes. For example, here you will have the name of that sequence. So this is the NCBI ID and the name of that organism, including a full sequence without breaks of just nucleotide letters. Here you have the same, so you can see that NC underscore something, that is for nucleotide, and then a virus, and this is the complete genome. This is what a FASTQ file looks. So a FASTQ file has the name of the read and then has the sequence of the read. So this is some characteristic of the read, so length, for example, etc., and then the actual sequence. And then below it, you also have information about the quality. So here you have the read, and for each one you have a score for the quality of sequencing. Now, this is the process how these types of data are generated. First of all, the genomic DNA is sheared or broken up into small pieces. This could be done mechanically or enzymatically, so you can have specific enzymes or you can just break it up. And those become small 200 to 300 base pair fragments. So these fragments are then used to create what's called a sequencing library. Typically, that is done to select for a specific type of DNA or RNA that is present there. And that information is applied to a flow cell. So it's included into a flow cell, which is inserted into the machine with PCR amplification. So what the machine sequences is the actual reads, the actual fragments, including these adapters, and after amplification. So oftentimes that is done because there's just not enough genetic material in the source sample. But as a result, what the sequencer does is it looks at different colors that become visible after an enzymatic reaction, and the imaging allows us to identify with different levels of quality what is the output in those 4 different nucleotide options. So as a result, we have this sequence for each one of the reads. Okay, so what can we do with this type of information? So once we have those reads, what can we do with those reads? One of the major objectives of different types of analyses is to identify variation. So if we want to think about identifying variation, we obviously have to know where these short sequences come from. And once we know where they come from and they are aligned, so they are positioned to the correct reference on the genome, we can then start asking the question, for example, what might be the result of finding a particular mutation, a particular variant? After we've identified those variants, or after we have compiled a list of those variants, we can think about how to interpret them. Another approach is when we're not sequencing to study variation in a particular position, but instead looking at how similar or different are different genomes or genes. So here, for example, you can see multiple sequence alignment of genomes, and those genomes help us understand relationships between different groups of samples that we have, called phylogenetic analysis or evolutionary analysis based on multiple sequence analysis. So we'll talk today primarily about alignment and identification of those variants, but also you have to keep in mind that there's many applications to what you can do with that in the downstream, the next steps after that. So obviously, when we talk about identification of specific variants, not only is it important to identify the actual change in the nucleotide, but also then map it onto those codons to identify the impact. And we can oftentimes compare and get very different results when we look at the amino acid sequence or the codon sequence, as well as the nucleotide sequence. And we'll see that in a few minutes when we do the practical hands-on assignment. Now, phylogenetic analysis, which is applied oftentimes when you have different genomic sequences from viral, bacterial, or individual genes from humans or other species, you can build a phylogenetic tree, and that phylogenetic tree allows us to study how different sequences are related to each other instead of looking at specific positions that are changed and thinking at what kind of change that might mean in terms of protein structure. So this is more of an analysis of how they are related to each other evolutionary, and that's what we call phylogenetic analysis. Oftentimes you can do this kind of phylogenetic analysis with viral sequences, and what it really does is it helps us identify relationships and then focus on specific changes. So when we talk about specific changes, oftentimes we are looking for SNPs, or single nucleotide polymorphisms. These single nucleotide polymorphisms emerge as a result of alignment of reads onto the reference genome and the process of annotation. So what is the difference between a mutation, a variant, and a single nucleotide polymorphism? So that's a question to you. What do you think is the difference between a variant, a genomic variant, a single nucleotide polymorphism, and a mutation? Does anyone know what is the difference? Okay, so Susanna is answering the SNP has to be present in at least 1% of the population. Okay, what else do you know about SNPs, mutations, and variants? Any other guesses? Okay, so a variant is what we see right here. So this is what we identify in the specific reads that are aligned to the reference genome, right? It's just a variant. So it's just a variation. It's different from the reference sequence. A SNP is a variant that occurs in a population. So if we talk about humans, for example, single nucleotide polymorphism is something that is observed frequently for some percent of the population, perhaps at least 1% of the population. A mutation is typically a SNP that is associated with some kind of a phenotype. So not all SNPs are going to lead to a change in the phenotype, but if we know that it leads to cancer, or we know that this is associated with some kind of a process or a phenotype, we can call that a mutation. So as you can see, the obvious challenge here is to look at how confident are we that we are actually finding a SNP and that that SNP or that variant leads to some kind of a change. For example, we can have, here you can see an alignment of reads. The number of reads that we have for each position is called coverage. And when we map it onto the reference, we have different levels of confidence in what we're actually finding. For example, this first position change from T to A is a substitution because it is exactly the same in the majority of the reads or all of the reads that we have for that position. Here we have at least 2 reads that indicate that we have an insertion or a deletion. In this case, we have a deletion. So we have a number of reads that indicate that we have a deletion. But here we have something that is very difficult to characterize. For example, we have just 2 reads that show that something is different, but we don't really know what is different. And so when we talk about this process of analysis, that includes multiple steps that have to be addressing this challenge of making sure that the read is properly prepared for analysis, then looking at accurate mapping or alignment to the reference genome, the ability to call a variant to really identify the difference between ambiguous changes in the sequence and specific changes in the sequence that we can rely on. Sometimes we want to use that information to compare different types of sequences. And then there's an additional challenge in annotation, which requires access to databases and different types of information that comes from outside of the actual sequence that helps us interpret and compare whether such a position has been observed in other types of sequences, and potentially if it is also found in association with a specific phenotype change. Now, there's also a step that we can apply that's called post-processing, and post-processing helps us evaluate the different types of information that we have through the processing steps to understand whether there's an enrichment in mutations, for example, for a particular segment of the genome, or if there's an accumulation of mutations in closely related positions. That relationship could be identified statistically as being a part of a consistent region or regions that might be interrelated or similar. Okay, so any questions about this so far? Does anyone have any questions or comments up until now? Okay, so if there are no questions, let's go ahead and do this together. We'll do a little practice run. So please go with me to server.t-bioinfo and let me know once you are there and logged in. How do you know that you have logged in? You will see your email address pop up right here under this icon for your profile. So please let me know when you're there. Thank you, Prashant. Susanna. Okay, Irmak. Okay, thank you, Simon. All right, so here we have different types of analysis. Let me just make this a little bit bigger. And we'll do a short demo today just to kind of understand how things go. And after we do what we do today, you will have the ability to actually upload some files and see some of the settings as we go through Genomics 1. So in this next generation sequencing data section, we will go to mutation variant, parallel analysis of mutation variant data. So if you click right here on the down arrow, you'll see here a demo. So let's go right here. and open up this section for mutation variant analysis, parallel analysis of mutation variant data. Let's click on Start, and we'll see here that mutation variant analysis typically offers us an insight into single nucleotide variants, often referred to as SNPs. So some of the things that we already talked about today. And let's first build a pipeline. So we'll just align our reads. So the Start button is the button that loads the sequence into the pipeline. And we can go to bowtie 2, which you can see is a method that is efficiently mapping the reads to the best position on the reference genome. So the way it does it is by hashing and doing all kinds of possible rotations of the reads and then picking the best position onto the reference genome. So we have an alignment and we have the quality of alignment. Now, as a result of this step of bowtie 2, the bowtie 2 algorithm produces a file called SAM or BAM file. So if you look at the result here, you will see that there's an actual file that is being produced, and that file includes the position and the end of each one of our reads. As a result of this process of alignment, we can visualize the SAM file. So the SAM file that we'll look at today is going to be visualized in JBrowse. So we're just going to do a simple pipeline like that, and then click on End. And after the pipeline is done, you will see a link here for visualization. Okay, so let me know if you have gotten this pipeline completed with visualization results, and we will look at some of these results right here. So how many of you have gotten the result the same way that I'm looking at it? All right, great. Okay, so what do we see here? First of all, we see here, let's forget about the reads for a second, and just let's look at the coverage. Okay, so you can see that we have two samples, SRR 925765 and 925766. And the first thing that we can see here is that they have different coverage, right? So one has coverage of about 128, and the other one has coverage of about 439. So you can see that right here. If we zoom out, we can see that the coverage is not consistent throughout the whole genome. So just based on what you see here, what do you think, what kind of sequencing do you think this is? Would this be whole genome, whole exome, or targeted sequencing? And you can zoom in and out to determine that. So what kind of sequencing do you think this is? Yeah, so Simai is suggesting that this is exome. Why do you think this is whole exome? Okay, so what specific regions, and how do you know what regions do we have here actually? All right, excellent. So coding regions. So how did you determine that these are coding regions? How do you know that these are coding regions? Okay, yeah, so right here on top, we have the GTF file. So this is actually what's in the GTF file. So the GTF file provides us with information. We can click on it, And you can see here all of the information about this particular sequence. So what is this sequence? This sequence right here is a part of a gene. Okay, so you can scroll down here. And this is the mRNA, right? So the mRNA is a transcript. So ENST is a transcript, and this is the name of this mRNA, and it belongs to this gene called P53. So P53 it's the transcript of the P53 gene, and that's a particular variant, because if you look here, there are multiple. So there's TP53, BP2, 1, 2, 7, 5, 10, 14. So it has a lot of isoforms, a lot of transcripts. So these are known transcripts that are expressed or transcribed from this gene. And so you can see that the coverage, right, is aligned pretty well with those red areas, which indicates that these are the exons of this gene. So all of the introns are black, all of the exons are either red or gray right here. And you can zoom out a little bit just to see that all of our data kind of groups around these individual exons. Now, coverage within those exons allows us to study mutations or variants, right? So let's take a look at how we can find those variants. So if we zoom in, we can see, for example, right here, right, we have this gray marker at the bottom. Okay, we have this gray marker, and the gray marker indicates a variant. Okay, but here it's not very obvious what this variant is, so we need to turn on the second part that actually shows us individual reads. So here we have a position, right, and now we can look at what is within that position. So 765, we can see that the T is present in all of the reads, right? And if we look at the second sample, which is 766, it has T in about half of the reads. So here we can be confident that a mutation or a variant is present in all of these reads. And we know that these reads right here, you can see that about a half of them, if you just hover over, it says that the in about 41%, this T is present. And here you can see that it's present in 97%. Okay, also you might notice that some of the reads are blue and red. So the blue and red indicate the positive and the negative, so two opposing directions. And the reason for that is because this data is what's called paired end sequencing. Paired end sequencing means that each read, each short fragment that we have from DNA is being sequenced from both ends, and that allows us to be more confident that what we find in the sequence of those reads is an actual non-technical variant, right? So if we have some kind of a mistake, it's more likely for that mistake to happen on just one and not the other. But if they happen in both, we have higher confidence that that's actually a position that we should be mindful of. For example, you can see here reads that are positive and negative direction, and they have very different information in them. So for example, this one has a lot of variants. Probably we're going to just ignore them because they actually do not happen at the matching read from the other direction. Okay, so is it clear how we can read this information? So you can actually click here, you can see the sequence of the read, you can see the length of this read, so you can actually see what this information, where this information comes from. Which color is reverse? So you can see here, okay, so right here, this is the positive. So if you click on them, you'll actually see. So right here, you can see that all of the red ones are the positives and all the blue ones are the negatives. Okay, so just explore the data and try to see what are all of these colors. So what do they all mean? Okay, and the ones that you see don't match, they will be colored dark blue, well, dark red. Okay, you can see multi-segment all correctly aligned, no. Okay, so it doesn't have a complementary one. Okay, so you can really explore this data and navigate between different positions. Prashant, yeah, we might be accessing it all at the same time, so it might take some time for you to load. Again, remember that these are huge files, so it is going to be based on your connection as well. Okay, so here you can also navigate between the different regions. This is chromosome 1, and you can see all of your chromosomes listed out here. You can also search for a particular gene. So for example, if I want to find the BRCA gene, and this is an example from breast cancer, I can actually find the BRCA gene right here just by searching it in this field right here. Okay, and you can see here as well, you have the reference, and you can see here's a large exon, and here we have a lot of coverage. Okay, and so here you can see that the coverage is actually more comparable. This is 500 or around 500 at the highest point. This is about 550. Okay, so any questions about what we did here with visualization? And apart from Prashant, was everyone else able to load the data and explore the data in your browser? Please let me know in the chat. Okay, so please try this after the session is over. Try to do this independently. Again, this is a demo, so you should be able to repeat the steps that we have right here. And after you've repeated the steps, you just click on visualization and explore it. And now I will show you an additional way to study this data. So if you want to, yeah, maybe the connection is not that good and you also have Zoom on, so it might be limiting your bandwidth. Okay, so let's take a look at another demo. So you can just click here, create demo mutation variant pipeline. So let's try this together as well. And then let me know if this works for everyone to download the files instead of trying to do this interactively right now. So please come with me to create demo mutation variant pipeline. We'll do another type of analysis just to understand what other outputs are possible. Let's click on start. And then we'll clean the reads from PCR amplification. We'll again map those cleaned up reads onto the reference genome. And then we'll do what's called differential variant calling. So differential variant calling will allow us to find the positions in the genome that are different between a tumor sample and a normal sample. In cancer, typically we are looking for somatic mutations, mutations that occur as a result of replication of cells. And that is done by comparing the tumor sample to a normal sample to the reference genome. So it's kind of like a three-step process. And we have two examples here, Strelka and somatic sniper. So these are just two different approaches to calling those variants. And then we will click on end. So as we saw in our example previously, some positions have 100% change from the reference genome. Some will have that change in one of the samples, but not the others. And in some, there will be some percentage of reads that has the change and some percentage that does not have the change. So that's what variant calling really comes in. It starts looking at different types of combinations and produces a report. And so let's take a look at the report. So please download this file. And let me know once you've downloaded the file. And extracted, obviously. And let's just take a look at what's inside. So inside you will find, actually, I'm just going to delete this because I have this from yesterday. Okay, so here inside you will find two files, cell line somatic sniper, an Excel file, which all of us should be able to open. So let's open it up. And for those of you that will continue for Genomics 1, I recommend downloading IGV to explore the VCF file. So we'll do that in our session next time. But for those of you that want to analyze and look at this data on your computer, it will require some memory on your computer because you have to have at least the human genome, which is, I think, 3 gigabytes or over 3 gigabytes. But this is something that will be useful, and you'll see how to use it in Genomics 1. So let's take a look at this Excel sheet and try to understand what we have here. So you can see that for each position, we have the chromosome, the position on that chromosome, the reference. This is IUB genome type of tumor and IUB genome type of normal. So we have a comparison between the normal. So you can see that the normal matches to the reference, but this one does not match with the reference, right? So that means that the tumor genome has a mutation, has a change. So as you go through here, you can see that there's also a somatic score, and that score, that score is what allows us to distinguish between a low confidence and a high confidence change. So here you can see highlighted are all of the ones that have above 200. So if it has a score of above 200, we have a high confidence that it actually has been detected in significant numbers of samples of reads, within that sample. So really, if we want to find only the positions that we would like to study for annotation to understand which gene they come from and what exactly might they mean, we can sort this. So we can just filter this and say anything above. Okay, so here, for example, I can do just by color, but you will see that all of these are above 200. So all of the highlighted are above 200. And so I have here 1, 2, 3, 4 important mutations for me to look at. So again, it's depending on your threshold. This was highlighted just for the example to talk about these mutations, but obviously when you do your own analysis, you will be able to set the threshold yourself. So this is just an example that we're going through to explain how this works. So here in this example, we're just taking these 4 mutations to be able to look into them and try to understand what they mean. Does that make sense, Irmak? And obviously you can change this to, instead of being the highlighted, you can select a threshold that seems meaningful to you. And that's going to be based on the coverage and the understanding of the analysis itself. And based on that, you might want to change it to, let's say, 201 or something like that. Yeah. All right. So I hope that this was important for you to understand as you look at the different types of changes. So you can see here also you have an annotation of what each letter means. So these are the different codons. Yes, in this specific output, you only consider this one, which is the somatic score. Okay, and what you will have in the VCF file is based on this threshold. You will just have these positions and what exactly is the information for each one of your positions, which is included right here. So that is the difference between the raw output, which is an intermediate output, and the final output, which will be the VCF, the variant call file, which only includes these positions on these chromosomes, and you can visualize them in the integrated genome viewer. and then annotate them based on different types of databases that are available there. Okay, so again, for those of you that will continue exploring this type of an analysis, which I recommend to do, and you can do this by going through Introduction to Genomics and Genomics 1, you will be able to visualize the VCF file in the integrated genome viewer. So we won't do that together right now. And you also have to remember that there's a number of different options for this kind of an analysis. You can do variant calling, and you can use here a model or a non-model genome. For example, if you take a non-model genome like Ebola virus, you can still map your reads onto the Ebola virus, and we'll talk about just a couple of examples right now. Or you can do model genome, and this is differential variant calling. And so obviously when you do this pipeline yourself, you'll also learn about settings when you upload your files. And when you upload your files, you have to determine whether you have just something that is mapped onto the reference genome for germline or just simple mutations like in viruses and bacteria, or if you are looking for somatic mutations, you might want to look for group treatment and control, compare them to each other and to the reference genome. So let's talk about some examples that we'll look at. Cystic fibrosis. Cystic fibrosis is caused by a mutation, a germline mutation. Cancer is caused by somatic mutations and some germline mutations as well. A good example is BRCA. The BRCA gene carries a risk, and depending on whether the parents both carry that mutation or not, the child has a greater or a lesser risk of developing breast cancer in their lifetime. And so you can screen for those mutations to determine the risk. You also have other applications for variant calling, including tuberculosis. For example, drug-resistant or multiple drug-resistant tuberculosis is an emerging danger around the world, where tuberculosis cannot be effectively treated with multi-drug therapy, and those strains of tuberculosis that emerge have to be quickly detected to prevent infection. And then a good application for this also is Ebola virus, and we'll talk about this type of mutation analysis that also allows us to do machine learning and different types of advanced analyses to really understand composition of different viral pandemics or epidemics. And we can do the same, of course, with COVID or other types of emerging pathogens to really understand whether there might be a relationship between accumulating mutations in viral sequences and viral genomes and phenotypes like transmission rate, infection rate, the mortality rate, and other types of parameters that we can measure. So let's talk about some of these biological examples. Here we're looking at cystic fibrosis. So cystic fibrosis is a result of a mutation in the CFTR gene, which is in chromosome 7. And you can see that different types of mutations, so here you can see the actual mutations, right? So these mutations are known to cause a particular type of a phenotype. So here you can see the percentage of patients with this type of mutation. So there's 22% of all cystic fibrosis patients with this particular mutation. This is going to be your position within that gene. This is going to be that position. And this is what the mutation is. So for example, this is a deletion and this is a change, right? And here you can have even a change from C to T. So different types of mutations, these are well characterized and annotated, could be resulting in different types of phenotypes. And here you can see that the implication of cystic fibrosis is the mucus that accumulates in the lung, because the channel, the CTFR gene, produces this protein, and this protein is responsible for pumping out the mucus. And so when it doesn't, you have this accumulation of mucus in the lungs, and it's a dangerous condition. And there's not really a way to treat it that effectively because this is a germline mutation. In other words, it's passed on from parents to their children. Another example is DNA aberration. In Huntington disease, for example, it's not a single mutation, but it's actually accumulation of specific types of residues in the Huntington gene. So there is an accumulation here. You can see 36 glutamine residues at the end of this specific sequence. And so as a result of this accumulation, which can be seen here, these are all of the chromosomes. So you can see that 1, 2, 3, 4, 5, etc. So these are all the chromosomes. And here you can see an accumulation of these mutations. And that is something that causes Huntington disease, which is a neurodegenerative disease, which was discovered by whole genome sequencing. So here, this particular figure is from a 2015 study by the Huntington Consortium that published a study of over 1,000 individuals with early and late onset of disease. Applying targeted sequencing as well as whole genome sequencing, so combining two methods of sequencing, the researchers discovered important regulation mechanisms that differentiated between these groups, providing insight into other mutations, as shown here on chromosome 15, that were associated with slower development of clinical onset. So even though the mutation might be present, the speed at which the disease progresses could be different between patients. And so you can really combine germline mutations, so with whole genome sequencing identifying different types of other variants, and with targeted sequencing determining that there is this amplification of the glutamine chains. In cancer, we talked about somatic mutations. So these are acquired mutations, which are the most common cause of cancer. And they typically occur in tumor suppressor genes or oncogenes. So for example, tumor suppressor genes are genes that are responsible for fixing mistakes that happen as a result of cell division. And so those could lead to DNA damage, and those could lead to apoptosis in a normal cell like P53. However, in tumor cells, that control mechanism doesn't work anymore because of some tumor suppressor gene mutation. Oncogenes, typically there are a lot of genes in the body that could lead to overexpression of particular proteins or different types of phenotypes to the cell, and those are called oncogenes because if they are non-controlled and activated, they could lead to serious change in the phenotype. And so those are typically suppressed. But if they accumulate mutations and they become functional and they start changing the way that the cell behaves, those are called oncogenes. And so here you can see how those somatic mutations are hypothesized to accumulate, and you can study those mutations looking here at the different types of analyses that we just did. So you can see some of those examples, and in Genomics 1, you'll be able to open up the VCF file from Strelka that we just created and identify those mutations in P53 and learn how to categorize those mutations by annotating and understanding whether there is a known phenotype associated with those mutations. Some other applications you can see here for tuberculosis. For example, we can visualize and also study differences between different samples, leading us to discover how we can annotate these different types of tuberculosis drug resistance or multiple drug resistance mutations. And so here you can also see some genes and some mutations in codons that are responsible for this accumulation, and a study of the Ebola virus. So in the Ebola virus, again, we can look at accumulation of mutations, and that accumulation of mutations actually has led to an important vaccine that was developed based on some of the proteins that are important for the virus entry into the cell and effective replication. So I hope that today's session gave you a perspective on how genomic data is generated and analyzed to produce important information relevant to basic research and different types of biomedical applications. I hope that you also understand some of the challenges that come with next generation sequencing and the approaches to analysis. And as you explore in Genomics 1 and other courses that we will do in this program, you will also see how the same type of sequencing And the same type of analysis techniques allow us to study different types of biological phenomena and try to interpret it from what we already know and what we are able to extract from this type of sequencing data. Are there any questions about what we covered today? Okay, thank you, Prashant. So again, what we covered today is this section on Introduction to Genomics, DNA Variants and Mutations. Please try to complete Introduction to Genomics and Genomics 1, and then we will continue speaking about transcriptomics, so how we can study gene expression based on analysis of next-generation sequencing data. If you are already done with Introduction to Genomics and Genomics 1, you can go ahead and start with Transcriptomics 1. We will cover the process of how data is generated, analyzed, and interpreted biologically based on mRNA expression or expression of genes and isoforms. If you have any questions or trouble completing some of the assignments or what we did today in the session, please refer to the forum and post your questions there. We will be addressing those questions as soon as we get the time to get to the forum and see what the problem is. Thank you everyone for joining today. I invite you to join us later on today in about half an hour. We'll have a session on the launch of our coding platform. For those of you that are interested in coding, we'll review one of the methods of analysis that is going to be very practical for those of you that want to deal with genomic data. And so Vipsa, if you could please share the details of that session. And also we invite you to continue to build up your profile, as I mentioned before, so that your profile is complete and you can share your progress with others in the team. Would I recommend to finish the genome course before the metagenomics one? Not necessarily. If you're interested in the introduction to metagenomics, you can also go through that one. I think that relevant to today's session is introduction to genomics and genomics 1. All right, excellent. Thank you everyone for joining, and I look forward to seeing you again soon. Thank you, Bibsa.
Thank you, Ilya. Yes, so this is the link again. So we'll be starting at 11 a.m. CST today, if you're interested. Thank you for joining.